Good morning, everybody. Happy New Year. Hope everybody had a great holiday, got rested, ready to dive back in and finish up this year of biology on top. Oh, Andrew, you got a new mic. Colorful. Ooh, that's cool. All right, anybody do anything interesting over the break? Nobody? Nobody's sharing? I'll share. I did go to the beach. I saw dolphins swimming. I did not ride a horse, but I went to a ranch and I fed a horse. Fed some horses. Fed a very mean horse. I did go to the beach. I did have a good time. I walked every morning. A couple of mornings it was cold. One morning I walked to the beach. It was too cold, and I went to the gym. And the gym looked at the beach. It was it was definitely a cold holiday. But other than that, it was a nice beach. Like I said, I went to a ranch where they had horses. I didn't want to ride horses because my thing is I want to ride a horse on the beach, and these horses that were close by were only took you on a trail, and I didn't want to go on a trail through the woods, so I just went and looked at the horses, fed the horses. They had other. It was like a petting zoo also, so they had llama and goats and sheep and all that kind of stuff there too. So that was interesting. And then they had the place where I stayed, they did two presentations. One was on different days. One was on the wildlife in the area. So I learned about alligators and stuff too. So I know how to stay away from those. And then they did on another day, they did birds of prey presentation. So they did three types of owls. So they actually had the owls uh, in the room. They walked around and so that you could see the owls up close. I got some nice pictures of owls and a very large red-tailed hawk. And so she would every once in a while she'd raise her hand so that he'd spread his wings, and he was quite big. So I learned a lot about birds and owls and things like that. So it was interesting. It was interesting. Then family came eventually. I spent the first couple of days by myself, and then family came. And so it was a nice holiday. Very nice. And then I came back home and it was all rainy and dreary and cold some more. And that was no fun. All right, I think we got everybody. Everybody's here. Good, good, good. All righty. Well, um, as far as your notes from last semester, keep them in a folder somewhere because again, this is a end of course milestone course. So you want to keep them in a folder somewhere that you have access. Of course, you have Canvas and you can always go back and read all of the chapters in Canvas, but some of the handouts and worksheets that we did, you want to keep those handy, don't delete them, so that you have them to look back on and study for the milestone, because that is in April. We did the benchmark in December, and most of you did very well. But you will see that benchmark, that same benchmark again in March, so that we get an idea of your progress. Again, it's not a grade that goes in the grade book, but it does let us know your progress. And at that time, we kind of set up tutorial sessions to help you make sure that you're successful with the milestone exam. So um, taking that test again in March will give us indications of how to help you best do that. Okay, so again, stay focused. This is a class that you want to pass. You have to pass it to graduate. Otherwise, you got to take it again. So you want to make sure that you stay on top of things, try to do things in class when class time is provided, and make things turned in by the due dates. Uh, and that's all the preliminary stuff I want to say. We're starting cells. We've done cell structure before, so we've talked about organelles and all this. And then the thing that we left off on last was DNA and DNA replication. So that was just looking at the DNA. So now we're going to look at the DNA in the cell. So we're going to refer to DNA replication, but we're really going to be looking at how that dictates how the cell as a whole will replicate. Okay. So look at everybody log into Canvas. Everybody log into Canvas, and we look for the unit on cell reproduction overview. Now, this one, I'm going to tell you that this one is kind of a lot. Okay, this is not an easy unit to get through. There's going to be diagrams and things that you're going to have to match up information, and I'm going to guide you through those things as we go through today. There are 4 sections to cell reproduction, or 4 different kinds of cell reproduction: mitosis, meiosis. We're going to talk about asexual as opposed to sexual reproduction, and then cancer, because what cancer really is cells growing out of control in places where they shouldn't be growing at all. Okay, so we're going to look at, as we get to how cells reproduce, then we're going to look at what happens when things go wrong. So again, when we did DNA replication last time, we talked about mutations, and there was a section of mutations, chromosomal mutations, that we did not talk about, and we're going to talk about them now, because we're going to look at what is a chromosome, and how we have different genes. There'll be a couple of activities where you're going to look at identifying genes and knowing what genes do what. And as you can see, there is a long list of vocabulary words. Okay, and some of them sound alike. So you're going to have to be real specific about what you're doing. Like, we're going to talk about chromosomes, and then chromatin, and then centromeres, and all these things sound alike. So you're going to have to kind of keep referring back to your vocabulary list to know which one is which. Okay, so I want you to give a look. In fact, I'm going to stop sharing, let you look that over for a few minutes. And since everybody's here, let me do the attendance real quick. Okay, and so you're going to the restroom, go ahead. And I'm going to do the attendance. And it's nice that everybody's here on time. We can get started. Okay, and then after you look at the overview page, just flip over to the first section, because we are going to start with the first thing on that list, which was mitosis. So again, look at the vocabulary. I'm going to do attendance, which will be quick because everybody's here. Um, Alrighty. All right, so again, everybody should be logged into Canvas, and you should be looking over that first page of the unit, especially the vocabulary, especially the vocabulary. Right, and again, I do want to repeat that if you are in class, use class time to do class work. Should be someplace else. All righty, back over here. All right, mitosis. So again, on that first page, me come back. First page. All right, so I do want to emphasize again that this particular unit is a lot. This is not a unit you want to be taking a nap and doing other, trying to multitask and all that sort of thing, because it's a lot. We are live streaming, and I think I figured out how I get in trouble all the time with doing things that I know are okay, but I think I've got it so that they won't lock us out. So we are live streaming, and the videos for today's lesson are in the playlist already. We're going to watch two of them. So the two things that we're going to look at is a general definition of cell reproduction, and then we're going to look at what is mitosis. Then I want you to have a pen and paper handy, because I do want, there's a point that I'm going to stop and tell you to take some notes. Okay, we don't do that often, but you're going to want to take some notes on this one, because you're going to need to create a script for you to look at, so that you can, your personal little cheat sheet, so that you can match up pictures. You're going to have to match up a picture with a description of what you see in the picture, and you'll know what I mean when we do it. But we're going to do mitosis today. Okay. Alrighty. And I'm going to go ahead and show these two. All right, this lady, she gets real deep and intense, and so you can watch her, but I don't find her to be as clear as you can be. So you can watch. I'm going to show you a different video. So we are going to talk about cell division. Cell reproduction is also called cell division. And before we look at it, we're going to watch a kind of a review of what we did with DNA. So that's what this one is.
Hi there. Let's discuss cell reproduction. Before we start, let's begin by reviewing DNA. DNA stands for deoxyribonucleic acid. It's a type of nucleic acid, which is one of the 4 macromolecule groups. When a eukaryotic cell is not reproducing, its DNA is inside the nucleus as disorganized long strands called chromatin.
All right, I'm going to pause it there. So we have to remember this nice long word up here, eukaryotic cells. Eukaryotic cells are the ones that have a definite nucleus. When you look at the cell, it has a membrane around it, and all of the DNA material is inside that structure. Then we have prokaryotic cells, which also have DNA, but they're not inside a nucleus. So you do need to know the difference between a prokaryotic cell and a eukaryotic cell. We are going to spend most of our time talking about eukaryotic cells, because we have eukaryotic cells. So most of the discussion is about the process that goes on in eukaryotic cells. We will have a brief section on prokaryotes, and she's going to touch on that, or one of the 2 videos we're watching are going to touch on it. But most of what we're going to do is in a eukaryotic cell.
Before a cell divides, the chromatin thickens and shortens into distinctly visible bodies called chromosomes. Each chromosome is a single DNA molecule. In order for a cell to divide, its chromosomes must first make a copy of themselves. This is DNA replication, so that each new daughter cell gets the original amount of chromosomes. Just after chromosome replication occurs, the 2 chromosomes, old one and new one, are stuck together. These 2 structures are referred to as sister chromatids. They are joined together by a central protein bundle called centromere. The sister chromatids are exact copies of each other, and each one will be distributed to one of the new daughter cells. Now that we have reviewed DNA, let's discuss mitosis. Cell division is the production of creating new cells. Cells divide in order to grow or repair damaged cells. An example is your skin or intestinal lining, which divide regularly to grow in size and or repair tissues. Not all cells divide though. A number of cells in your body, such as nerve, red blood, and muscle cells, do not divide once they are mature. The cell cycle is a timeline of events that occur during the life of a cell. The cell cycle is composed of mitosis and cytokinesis. The cell spends most of its time in interphase, which is the green area. During interphase, the cell is getting ready to divide. Interphase is divided into three sections, G1, S, and G2. In G1, the proteins are produced for the cell that will be necessary for cell division. In S, DNA makes a copy of itself, which is the DNA replication. In G2, cells make copies of the organelles. So mitosis is a type of cell reproduction that produces identical cells. This means producing cells that have the same DNA.
Oops, I was trying to catch that. All right, as we go through these, we have the two types of reproduction. Mitosis, it's important for you to remember this part, that they produce identical cells, that they have the same genetic material, okay? Because when we do meiosis, it's going to be different. So, and when the question is always, what is the difference between mitosis and meiosis? So mitosis is identical cells. The other thing that we are going to, you do have to know this, that we have the cell cycle. We're going to talk about this PMAT in a minute, and that's the mitosis part. But most of the time, the cell is in the green stage, which is just interphase. So we're going to talk about each of these separately. Okay, she was just about finished. So I'm going to go ahead. So we're going to come back and look at this right after we watch the video. And then we're going to go through each of these. I'm going to have you write these down. So this is the part you're going to write down, but we're going to watch the movie first. And I will pause it as needed. And again, please pay attention.
Good morning and welcome to Biology. I'm Paul Suchecki and this is my classroom. It's great to see everybody's smiling face again today. Today's lesson is on cell reproduction. You know how living things grow. You're a living thing. You still growing? Probably. I stopped growing a long time ago. Trees grow, grass grows. Oh, I just remembered. I gotta cut my grass this weekend. One way living things grow is by cell enlargement. If their cells get bigger, the entire organism gets bigger. Another way is by cell division, where one cell divides into two. 2 cells become 4. 4 cells become 8. 8 cells become 16. 16 cells becomes 32. 32 cells become 64. I think you get the idea. The more cells you have, the bigger you are, right? Think about an elephant. Think about a mouse. Does an elephant have bigger cells, or does an elephant have more cells? Are a mouse's cells smaller, or does a mouse just have fewer cells? Cells come from pre-existing cells. In other words, cells make cells. Cells make cells by a process called cell division. One parent cell divides into two daughter cells. I don't know why they're called daughter cells. I think they should be called son cells. Or at least let's be gender neutral and call them sibling cells. Anyway, nobody invented cell division. Nature invented cell division. And it's been going on continuously since the beginning of living things. Cell division is how multicellular organisms grow. It's how unicellular organisms reproduce. In a eukaryotic cell, the DNA is located inside the nucleus, and it takes the shape of a double helix. Think of DNA like a long thread that stores information. Most of the time, the DNA is just lying around the nucleus in a loose, unraveled form called chromatin that's wrapped around special proteins called histone proteins. I call chromatin the working form of DNA. When the DNA is in the chromatin form, the cell can read it, follow its instructions, and make any protein that the cell needs. Just before a cell divides, but after replication, the DNA is supercoiled into little bundles called chromosomes. Chromosomes are bundles of supercoiled DNA. Each chromosome has two halves. Each half is called a chromatid. The two chromatids are mirror images of each other. They're the product of DNA replication. They hold the exact same genetic information, or at least they should. The two sister chromatids are held together near the middle by a structure called a centromere. During cell division, the centromere breaks apart and the two sister chromatids are pulled into opposite daughter cells. Chromosomes always come in pairs, just like shoes, just like gloves, just like pants. Humans have 23 pairs of chromosomes for a total of 46. Pairs of chromosomes are homologous. That means they're similar, but they're not identical. Kind of like your shoes. What I mean is, let's say there's a gene on a chromosome right about here for eye color. On the homologous pair, there's also a gene for eye color in the exact same location, but it might not say the same eye color. One chromosome might say blue eyes, the other chromosome might say brown eyes. But the gene for eye color is in the same spot on both chromosomes. That's what I mean by chromosomes are similar, but they're not identical. You received one chromosome from each pair from your mom, and you received the other chromosome from each pair from your dad. So of the 46 chromosomes, 23 of them, one from each pair, came from your mom, and the other 23, one from each pair, came from your dad. And you're the genetic combination of both of your parents. It's kind of like taking a half a deck of cards from your mom and a half a deck of cards from your dad and shuffling them together, and then let's see what you get. That's you. Normal body cells are called diploid because they have the full number of chromosomes, that is, 46 in humans. We use the symbol 2N to represent a diploid cell. When I say normal body cells, I mean things like skin cells and liver cells and bone cells and muscle cells and, all of your body cells. Gamete cells are haploid, meaning that they only have half the number of chromosomes. Gametes are reproductive cells, sperm cells in males, egg cells in females. We use the symbol 1N to represent a haploid cell. I hope it makes sense that gamete cells are haploid. The sperm cell is haploid, 1N. That is, it has half the number of chromosomes, 23 in humans. The egg cell is also haploid, 1N. When a sperm cell and an egg cell come together to form a zygote, the zygote is now diploid. It has the full number of chromosomes, 2N. Before that zygote divides to form two cells, all of the chromosomes replicate, and then the zygote divides. That way, each daughter cell will have the full number of chromosomes. When cells divide, they go through a cycle called the cell cycle. During the cell cycle, there's an interphase between divisions. Some people call it a resting phase, but oh no, the cell's not resting during interphase. The cell's taking in nutrients, it's growing, it's synthesizing materials, it's replicating its DNA and getting ready to divide. I wouldn't call that resting. Interphase is the longest phase in the cell cycle, and it's divided into three subphases: G1, S, and G2. The G in G1 stands for growth. During the G1 phase, the cell's taking in water, amino acids, fatty acids, carbohydrates, and these materials are becoming part of the cell, which makes the cell grow. The second...
Actually, I'm going to pause it for a minute, just so that you do need to know what is the cell cycle. You do need to know what is going on during the G1 phase, the S phase, and the G2 phase, and understand that all of that is what the cell is doing most of the time is called interphase, because we are going to have to know the phases of cell reproduction. And then this little phase down here called mitosis that's we're going to spend a lot of time with that, but the cell spends most of its time in interphase, so you do need to know G1 is for growth, and then I'm going to let him finish. So if you want to take notes on that, you should take notes on that.
Part of interphase is the S phase. The S in S phase stands for synthesis. During the S phase, a duplicate copy of the DNA is synthesized by a process called DNA replication. The purpose of DNA replication is to make sure that each daughter cell gets all of the genetic instructions. During the G2 part of interphase, the cell's making its final preparations and getting ready to divide. The division of the cytoplasm and all of the organelles is known as cytokinesis. There's usually enough of each organelle so that when cytokinesis occurs, each daughter cell gets the organelles it needs. Of course, there's just one small problem. There's only one nucleus. So before the cell can divide, the nucleus has to divide. The division of the nucleus is called mitosis. Mitosis occurs in 4 phases: prophase, metaphase, anaphase, and telophase. Just remember PMAT. Sorry, Matt. Remember, we define mitosis as the division of the nucleus. Interphase is not one of the phases of mitosis. Interphase is the in-between phase, when the cell's not dividing. So by definition, we can't include interphase as one of the phases of mitosis. I've seen this error before. It's not IPMAT, it's PMAT. The first phase of mitosis is called prophase. During prophase, the nucleolus and the nuclear membrane disappear into the cytoplasm. The unraveled chromatin condenses and supercoils into chromosomes. 2 structures called asters or centrosomes appear at one end of the cell. One of the asters migrates to the opposite side of the cell. The second phase of mitosis is called metaphase. During metaphase, the chromosomes line up down the middle of the cell. There's got to be a way that you can remember that during metaphase, the chromosomes are lined up in the middle. Metaphase, middle, metaphase, middle. I'm sure you'll think of something. Also during metaphase, spindle fibers grow from each aster and attach to the centromere of each chromosome. The third phase of mitosis is called anaphase. During anaphase, the chromosomes are pulled apart. You see, the spindle fibers at opposite ends of the cell start to pull and play tug-of-war on each chromosome. This breaks the centromere apart, and the sister chromatids are pulled to opposite sides of the cell. Anaphase, apart. Anaphase, apart. There's got to be a way that you can remember that during anaphase, the chromosomes are pulled apart. I'm sure you'll think of something. The last and final phase of mitosis is called telophase. During telophase, the cell membrane pinches down the middle and forms what's known as a cleavage furrow. The cytoplasm is beginning to divide. Cytokinesis occurs, all of the organelles in cytoplasm are divided up, and we end up with 2 identical cells. The 2 daughter cells are now half the size of the original parent cell. In plant cells, since there's a cell wall, a cell plate is formed between the newly developing daughter cells. At the end of telophase, there are 2 cells. Telophase, 2 cells. You'll think of something. So what comes after telophase? Interphase, of course. The chromosomes unravel and become chromatin again. The nuclear membrane reappears and surrounds the chromatin. The nucleolus also reappears, and the 2 daughter cells go right back into interphase. They spend most of their time in interphase, growing, synthesizing, and getting ready to divide again. Remember the cell cycle? Prokaryotic cells like bacteria don't have a nucleus, but they still divide. Since they don't have a nucleus, and mitosis is defined as the division of the nucleus, we can't really call prokaryotic division mitosis. When prokaryotic cells divide, it's called binary fission. Prokaryotic cells don't have pairs of chromosomes. Instead, they usually have a single circular chromosome. During binary fission, this circular chromosome replicates, and then the cell simply splits in two. Compared to mitosis, binary fission in prokaryotes is relatively simple. The circular chromosome begins to replicate at one point in both directions. When the two replication forks meet, replication is done. Cytokinesis occurs, and the cell divides in two. Easy peasy. Meiosis, or meiosis, is it meiosis or meiosis? Anyway, meiosis is a special kind of cell division. Meiosis is the production of gametes, reproductive cells. In males, sperm. In females, eggs. Sometimes we call meiosis reduction division, because during meiosis, the number of chromosomes is reduced by half. In meiosis, we start with a diploid or two-end cell. The nucleus divides twice, and we end up with 4 haploid or one-end cells. See, the number of chromosomes is reduced in half. Reduction division. Why do you suppose gametes are haploid? Gametes have half the number of chromosomes. They're haploid, so that when they fuse together to form a zygote, the zygote is diploid. It has the full number of chromosomes. This does two things. One, it promotes genetic variety. The new individual is a genetic combination of both parents. But secondly, by reducing the number of chromosomes in half during meiosis, it ensures that the same number of chromosomes is passed on from generation to generation. In humans, that would be 46, 23 pairs. In order to reduce the number of chromosomes in half, during meiosis, the nucleus divides twice. So there's actually a meiosis 1 and a meiosis 2. During meiosis 1, there's a prophase 1, a metaphase 1, an anaphase 1, and a telophase 1. But that's just the first division. During meiosis 2, there's a prophase 2, a metaphase 2, an anaphase 2, and a telophase 2. When it's all said and done, we end up with 4 haploid cells. One of the main purposes of sexual reproduction is to promote genetic variety, genetic diversity. This is achieved a couple different ways during meiosis. During metaphase 1, instead of all the chromosomes lining up down the middle in a single file line, they line up next to their homologous pair. This is known as a tetrad. Then something interesting happens. Homologous chromosomes start trading little pieces of each other in a form of cellular genetic reshuffling that promotes genetic recombination and increases diversity. After crossing over, the homologous chromosomes are then sorted independently by a process known as independent assortment. In other words, the chromosomes are separated randomly, and this further increases genetic diversity. In males, meiosis occurs in the testes and produces millions of sperm cells every day starting in puberty. The production of sperm cells is known as spermatogenesis. In females, meiosis occurs in the ovaries, resulting in about 400 egg cells in a lifetime. These egg cells are formed in the individual's ovaries before they're even born, but they're not released until the individual reaches puberty, and they're usually released about 1 per month. The production of egg cells is known as oogenesis, or do you say oogenesis? Is it oogenesis or is it oogenesis?
Oogenesis.
During oogenesis, 4 haploid cells are produced, but only one of them becomes an egg cell. The reproductive organ for most plants is a flower. Usually, flowers have both male and female parts. The male parts produce pollen. Each pollen grain contains male gametes. The female gametes, or the egg cells, are produced in the ovary of the flower. Pollination occurs when pollen is moved from the anther to the stigma. The pollen moves down the style of the flower and into the ovary, where fertilization occurs. The zygotes develop into embryos. The embryo is hidden inside of a seed. Eventually, the seed falls off the plant, lands on the ground, and germinates into a new plant. So anyways, all living things grow, and one way that they grow is by cell division. You know, I asked you a question at the beginning of the lesson. Do elephants have bigger cells or more cells than mice? Well, it turns out that elephant cells and mice cells are about the same size. If you were to look at a little piece of liver under a microscope from an elephant and compare that to a piece of liver from a mouse, the cells would look the same. Elephants are bigger than mice because they have more cells, but their cells are about the same size. Anyways, that's about all I have for today, so I'm going to leave it there for now. Until next time, don't get lost and have a great day. See you later.
So that's an overview of everything that we're going to do. He covered all the vocabulary, and he covered both mitosis and meiosis. So what we want to look at today is just the meiosis part. And what I want you to do, this is where you're going to have that piece of paper to write it down. So hopefully I don't know why it jumps back like that. It's very annoying. Hopefully you stayed in your spot for, whoops, went too far. There we go. All right, mitosis. Everybody needs to be in mitosis because this is where you're going to write some stuff down. All right, the first thing you want to write on your paper is phases of mitosis. Like right here. All right, and then he mentioned the term PMAT. So you want to write PMAT next to your heading of phases of mitosis. And then what I want you to do is click on each of these, and I want you to write these things down. So I want you to write prophase. And the reason for this, again, you're creating a cheat sheet so that when you get the picture sheet that I'm going to send for you next class, you will be able to look at the picture and say, based on what I see in the picture, this must be prophase. Based on what I see in the picture, this must be metaphase. So you want to write this down. You're going to keep a handy description of what is happening in each phase. So in prophase, the chromosomes condense and become visible. And what they mean by that is when most of the time when you look at, and we're going to look at an actual, what they call a stained picture, and he showed them, but he didn't explain what he was showing you, what actual cells look like under the microscope. And when you're looking at a regular cell under the microscope, the nucleus looks very dark and dense, so you can't tell, you can't identify chromosomes. So it looks like a thick mass of spaghetti all wrapped up in one little ball. So he mentioned that it's kind of just wrapped around this thing called the histone. So what happens in prophase is they shrink. So our little spaghetti strands shrink and they become visible chromosomes. So those little X-looking structures that he was showing you, you begin to see chromosomes. So that's what's happening in prophase. So we can call, now he didn't give a word for prophase, but I'm going to call this preparation. Okay, so prophase, preparation. The cell is getting, the chromosomes are getting ready. All right, so they're going to condense, that means shrink, and they're going to become visible. Prior to that, they were just a squibbly little mass in the nucleus. All right, the second thing that happens during prophase is that the nuclear membrane begins to break down. So three things are going to happen during prophase. The nucleus is that envelope that's like that holds all of the stuff inside. But in prophase, when you're looking at a picture, you'll have a dotted line. It looks like a dotted line. So, and that's the indication that the nuclear membrane is starting to disappear. The third thing that happens is these little star things that he mentioned called centrioles. They are little rods, and most of the time in interphase, they're together. But during prophase, they move away from each other. So we call it moving to opposite sides, or we call it to the poles, like a north and south pole of the cell, or an east and west of the cell. And then they form spindle fibers, which are little fingers that start to grow out from them, and they grow to the chromosomes. All right, so there's three things in prophase. The chromosomes condense and you can see them. The nuclear membrane starts to break down and begins to disappear. And third, the spindle fibers, the centrioles move to the opposite side and start making spindle fibers. Okay, so hopefully you wrote all of that down. All right, next phase, and then come back and look at the picture too. So again, we're going to have, you will get a quiz that has pictures on it, and you'll be asked to look at the picture, say, what's happening here? All right, next one, metaphase. In metaphase is simple. The chromosomes, so now there's no nuclear membrane at all. It's gone. And the chromosomes are lining up in the middle. So that's what he kept stressing. You know that this is metaphase because the chromosomes are lined up in the middle. And the spindle fibers attached to them. So the copies of the chromosomes are attached in the middle by something called a centromere. So the spindle fibers will attach to that, which is important. And then we call each copy, and remember that it has replicated before it did anything else. It first replicated. So I mean, back up. These little X's, they look like little X's because they are the duplicated copies of the DNA. Okay, and the copies are held together in the middle by a centromere. So what these spindle fibers are eventually going to do is going to pull those copies apart. Okay, so each spindle fiber is attached, and they call those little X's, the copies, chromatid, and they're not chromosomes anymore. Each one is a chromatid, and they're sisters. There's no brothers. They're sister chromatids, and they are exactly alike at this moment. All right, then we go to anaphase. Anaphase is when the chromatids are pulled apart. So again, he kept stressing apart as your clue as to what's going on. So the spindle fibers are like rubber bands. They're going to shrink. As they shrink, they're going to pull those copies of the chromatids apart. Okay, hopefully you're writing this down. And I'm going kind of fast, but I'm going to stop and give you time to finish writing. And then the last phase is where a couple of things happen in the last phase. So now that they're apart and on opposite sides of the cell, the nuclear membrane starts to form around each of those sets of new chromosomes, and the rest of the organelles start to shift to different sides of the cell also. So that's PMAT, okay? So that's why there's a box around this, because PMAT, as he said before, these are the phases of nuclear division. So again, we defined mitosis as a division of the nucleus. All right, so after we've done that, then we're ready for the cell to divide. So that's cytokinesis. So during cytokinesis, the cell is going to split into two cells. In animal cells, as he showed you, it's going to pinch in the middle, and eventually the pinching will join, and then the two will just pull apart. In plant cells, they have a cell wall. So when we get to telophase, it's going to start growing a plate in between. So a plate will grow down. Instead of it pinching, a plate will grow down, and eventually this cell wall will, a new cell wall will show up between the two, and that's how you'll get the two new cells. All right, so I'm going to give you a chance to finish writing all of those down. Once you have finished that, I want you to click through this presentation here. It's kind of a repeat of what he just showed you in the video. This lady, again, she's going to go through what he just went through in the video. So if you want to watch that, you can. Like I said, she gets kind of intense sometimes. And then down here, you have another summary of what's going on. But I wanted you to write down the simplified one up top, so that, again, when you have a quiz, you don't have to keep jumping back and forth to look at what does it say in Canvas. But this is basically what I just asked you to write down. These pictures at the bottom are actual pictures, although I'm going to take you to a website on Friday where they are a little clearer. And we're going to look at the phases of mitosis that way. But these are what we call stained cells so that you can actually see what is happening in an actual cell. Okay. So when you finish writing, go through and read the rest of the page. And then when you're done, I want you to take, this is one of those drag and drops. So you want to, each side of the cell has a copy of its DNA. And so, and it won't stay if that's not where it belongs. Okay. So you want to go ahead and fill all these boxes up because again, this is a matching quiz part of the next test. All right. So like I said, finish writing this, come over here and play this video and read that over and then come down here and do the drag and drop. So that's what everybody's doing for the rest of class. All right. And please be doing it. Again, you want to practice makes perfect. And again, this box has the elongated version of what I asked you to write down. But again, you should study each picture so that you can see, relate to what's in the picture to what you've written down.
We we on the meiosis
So
part, right? Like
mitosis.
that's what we're learning.
Yeah, you're right. Writing mitosis. So we haven't gotten to meiosis yet. He was like, I say meiosis. So you'll hear me say it doesn't matter which way you say it. But we're
Yeah,
right now we're just doing mitosis.
I was just saying because I'm looking at my grades and stuff as well as doing the work. Are you aware like the meiosis, all that stuff is saying that it was due last year and it's charging my grade?
Oh, I didn't know that. So what
Yeah,
happened is we okay, I'll look at because it shouldn't. Okay, but I'll look at that. Let me stop sharing for a minute and go look at that. We haven't done anything. There shouldn't be any grades. So let me look at because I have not assigned anything for this year. The course was replicated from
yeah.
last year. So
It
all of the
yeah, it says
assignments
the
from last year are
meiosis.
there. Okay,
It
I
says
will
the
go in
meiosis
and
practice quiz and stuff.
okay, I will go in and no, I did not know that. So thank you for telling me that.
You're welcome.
Yeah. Well, okay, I see because you're looking at something, but when I'm looking at the grade book, there's nothing there. I got some question marks, but I'll go look at I'll have to go take the dates off of those assignments. Like I said, it shouldn't be any. But let me look. and clear that out. Because no, you have not done anything yet. Meiosis. Huh. I think I have to look under assignments. Okay. Okay. All right. I think it should go away now because I took off the published. So those should not have been published. I don't know why they said published, but they should not have been published. But no, those haven't been assigned yet. All right. So refresh your page and let me know if you're seeing the same thing now that I took off. Now that it's not published anymore. Is that better? Okay. Took it off there. Let me come back over to modules. Because my grade book didn't have any grades in it. Okay, good. And again, thank you for telling me that. Okay. Now one other one. Mm. All right, so again, everybody should be finishing up writing and then go down and do the practice drag and drop. Read the other box, get familiar with the pictures, and then goes down and read the drag and drop. You do not have to watch the video that she's showing you. Let me look and see if that's the same thing for the other class. Hmm, okay. I don't have that problem. How do you do the drag and drop? Okay. Let me come back and share. Okay. So with the drag and drop, you have the little green box up here that has a description, and you can use the box up top to help you figure out which one is which. And then you're going to drag that description into the box. Now, all the boxes have several answers. So it says each side of the cell has a copy of the DNA. So that goes into telophase. And you'll know whether it's right or wrong because it won't stay there. It'll pop back out if you're wrong. All right, spindle fibers are going to form. So that's what you're going to do. You're going to take as each phrase that pops up, you're going to drag it into one of these boxes. And when you're done, you should have, I think most of them will have at least 3 sentences in each box. Okay. Chromosomes are pulled apart. So again, apart. You're supposed to remember that A is, okay. Yeah, it's right up here. And apart is A. So that's what you want to do. Now they have an extra box here called prometaphase, and they do talk about it up here. When we do PMAT, we don't normally include this part, but prometaphase description is right up here. They have this other word called kinetochores. You're welcome. So again, if you get confused, pop back up here to this box and that'll help you. And again, just make sure that you read this one because it talks about the difference between plants, what plants do and what animals do. But they do mitosis. All right, so the important part about mitosis is this is how living things grow. This is how you get more regular cells. Okay. Okay, folks, time's up. So I hope you have a great rest of your day. And we'll practice some more on Friday. And we're going to look at some cells on another website called Cells Alive and do a little more practicing over there before we start looking at meiosis. Okay? Have a great day. Bye. Enjoy your lunch.